there, my name is Alex. If you don't pronounce the surname, don't worry, my wife can't either. And I'm a junior group leader in SBI, and rather selfishly, my previous speakers have introduced the whole center. I'm going to introduce something which is called systems biology, but it's only applied within my own group. So it's only a snapshot of what we're actually doing. And systems biology is something hard to understand for a scientist, and even harder for a lay audience. I'll try to package it so it's compact and easy to understand. And please start the slide. So what my personal group, we're looking at cancer cell migration. And the reason we're looking at cancer cell migration is a cancer patient generally does not die from the primary tumor. A cancer patient dies when the primary tumor spreads from the initial tumor and forms metastasis, as you can see from the image over there. And if we can't treat the metastasis, we probably can not heal the cancer, but we can prevent the patient from dying. So if you look at the molecular basis of what happens, cells start migrating or invading from the primary tumor, hitch a ride either on blood vessels or on the lymphatic system, and then spread throughout the body. So if we can prevent the initial step of the cancer migrating out of the tumor, we can prevent metastasis. So this is our general hypothesis, and we want to look at, on the molecular level, how a cancer cell acquires ways to invade the surrounding tissue and spread and form metastasis. And I hope there was another slide, yeah. So we're using a systematic way of looking at this. Systematic way means we're looking at all the available data that we can get. And one data set we got is from the Genome Project, and an analogy would be, it would be like the telephone book. A second data set we can get is functional genomics. So this describes the actual function or the job of the gene or of the protein it's doing. So we know where it's located, what it's doing. And secondly, now from biochemical data, we know what the gene needs to function properly, i.e. what it makes, what it buys, and what it sells. And more crucially, because cancer is a disease which arises from mutations, we're looking now at sequencing data which tells us if a gene is sane or insane or diseased. But what we want to do is want to look at the whole data taken together. So we try to integrate all the data and see how the individual genes or proteins interact in their own social environment. So we take all this data, integrate it, and when we try to make a mathematical model. So as I said, we take information from literature, from our own experiments or previous knowledge, and then we make a mathematical model which we construct in a computer. It would be similar to a model of a city. And then we take a look at the city and say we want to stop traffic. And we say, how can we stop traffic? We can, as an example, blow a huge volcano next to the city and see what happens to the traffic. And lo and behold, yeah, the traffic stops. Perfect. And then we imagine we are the gods of the ancient world, and we blow up Mount Vesuvius next to Pompeii and see what happens to the traffic. The traffic stops. Perfect. Manage that. But then something unexpectedly happens, so we didn't predict in our model that everybody dies. So we feed the data now that everybody dies, the city is destroyed, back into our model and see, is there another way how we can stop the traffic? And instead of blowing up Mount Vesuvius, maybe next time we'll just try to look at a bit of snow, or maybe the tube strikes or something like that. All right, and thank you very much, and especially for listening, and thank all the people for supporting.